This is a continuation of Chapter 2, Module 3, Organic Molecules. We will start with nucleic acids. Nucleic acids are the last type of organic molecule we will study. Nucleic acids are large organic molecules found in the nucleus of the cell. They store and process information for the cells. Two types of nucleic acids exist: the DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. The role of DNA is to determine inherited characteristics, such as hair color, skin color, or even how well one metabolizes medications. DNA also directs protein synthesis, controls enzyme production, and controls metabolism. RNA, on the other hand, controls intermediate steps in protein synthesis that you will learn more about in upcoming modules. Structurally, both DNA and RNA are strings of nucleotides. A nucleotide has three molecular parts: a pentose sugar, either deoxyribose for DNA or ribose for RNA, a phosphate group, and a nitrogenous base that is either adenine or A, guanine or G, thymine or T, cytosine or C, and uracil or U. Cytosine, thymine, and uracil are considered pyrimidines, and adenine and guanine are considered purines. Thymine will only be found on DNA molecules, and uracil will only be found on RNA molecules. The nucleic acid strands are long chains of nucleotides. Each nucleotide contains one base. These nucleotides are linked through the phosphate group on one nucleotide to the sugar group on another nucleotide through the process of dehydration synthesis. When several to thousands of these nucleotides are linked together, it makes one strand of nucleic acid. In DNA, two of these nucleic acid strands will be linked together by their bases with hydrogen bonds. Then these two strands that are linked together form a twisting double helix formation. When the nucleotides of one strand link to the nucleotides of the other strand, the bases that link together are called a complementary pair. The purines will link to the pyrimidines, so that adenine and guanine will link with thymine and cytosine respectively. In other words, adenine will only link to thymine, and guanine will only link to cytosine in DNA. DNA always has more than 45 million nucleotides in one molecule. DNA will store information that controls protein synthesis, or in other words, controls what, when, and how many proteins the cell will make. RNA is similar to DNA with some important differences. First, RNA consists of only one strand of nucleic acids, which means there is only one strand of nucleotides linked together through dehydration synthesis. The other difference is that thymine is not one of the bases on RNA. Uracil takes the place of thymine on an RNA molecule. And lastly, the sugar in the nucleotide is a ribose sugar and not a deoxyribose sugar. RNA may have anywhere between just a few nucleotides to up to about 50,000 nucleotides in its strand. There are three types of RNA we will study: messenger RNA, or mRNA, transfer RNA, or tRNA, and ribosomal RNA, or rRNA. RNA performs protein synthesis as directed by the DNA. We will talk more about DNA and RNA in the modules regarding the cell. Other than their use in DNA and RNA, nucleotides can also be used to store energy. Adenosine diphosphate, or ADP, is a nucleotide that contains the base adenine as well as two phosphate groups. The di in the word diphosphate tells you there are two phosphate groups. When a third phosphate group is added to ADP by using a high-energy bond to do so, the new molecule formed is adenosine triphosphate, or ATP. Adding this phosphate group to ADP is called phosphorylation. ATP is a high-energy compound that is basically storing energy in that high-energy bond between the last two phosphate groups. When energy is needed in the body, the phosphate group will break off and release the energy, which can then be used for whatever functions in the body require energy. We will learn more about energy usage in the body in a later module. This ends Chapter 2, Module 3, Organic Molecules.